Bright field microscopy, part 1. This is the topic of my lecture, and I am Sumati Hajela, Associate Professor, Department of Biotechnology, IPS Academy, Indore. Introduction. A microscope is a piece of scientific equipment that helps us to see things that are too small to be seen through naked eyes. The science of investigating small objects using such an instrument is called microscopy. So, first such instrument which we are going to uh, see today is bright field microscope, which is also commonly known as light microscope or compound microscope. And this is the simplest type of light microscope. The word bright field suggests that field around the specimen remains bright to give contrast to dark or colored objects. For cell biologists especially, it is the basic tool for studying cell structures. Principle. What is the principle on which why we can use this for studying small particles? So principle is like that. When the light rays are passed through a material, light absorption takes place. Contrast in the image is formed by differential absorption of light in various parts of the specimen. The specimen appears in a uniformly illuminated field, its structure revealed by varying contrast in color and intensity. There are many specialized techniques which have been developed for staining. They may be selective staining or differentially dyeing portions of otherwise transparent specimen so that they will exhibit different contrast and colors in bright field. Another part of principle is magnification by microscope. And what is magnification? It is an act or process of enlarging only in appearance and not actually in physical size. So the thing looks big, but actually it is not that big. Actually, it is so small that we cannot see that through our naked eyes. It is the measure of ability of a lens or other optical instruments to magnify, and it is expressed as the ratio of size of the image to that of the object. If we try to calculate magnification by the microscope, we will be using following formula, which is focal tube length divided by focal length of the objective lens multiplied by magnification of ocular. I will be discussing various parts of microscope later. Right now we are discussing is principle. So next part of the principle is uh, what is resolving power of this type of microscope. This type of microscope resolves up to 0.2 microns. And what is resolving power? It is the ability of an optical instrument to separate or distinguish small or closely placed objects as two different entities. That is, if two things are kept very close together, lens of this machine must distinguish them as separate entities. This is the power of lens present in the microscope. And if we try to calculate Resolving power, it is equal to the lambda by 2 multiplied by Na. So what is lambda and what is Na? On which resolving power depends? Lambda is wavelength of the light used and Na is the numerical aperture of the objective lens. Now, we know about the wavelengths. Uh, light, white light is made up of VIBGYOR, variety of colors and their shades. Basically, these are 7 colors, but their shades, in between shades are there. So every shade or color has a particular wavelength. So whatever wavelength we use, according to that optical instrument, uh, distinguishes small or closely placed objects as different entities. Numerical aperture, it is the measure of its ability to gather light and to resolve fine specimen details by working at a fixed object distance. It, its ability means objective's ability, which is in front of the specimen and gathers all the light to make image. If we try to calculate it, the formula will be Na is equal to eta sine alpha. Here eta is the maximum refractive index of various materials between specimen and objective lens, while alpha is the half angle of oblique ray that enters the front lens of the object. Light path of the microscope has been shown here. Source of light gives light which enters the condenser lens. Condenser lens focuses these rays towards the specimen. Light passes through specimen and due to differential absorption, various parts after colliding with the various parts uh, and their differential absorption, light rays enter the objective lens. Objective lens makes one image, which is again acts as new object for ocular lenses, and then this ocular lens makes an image which we collect through our eyes. This is the figure of or a picture of microscope, and I have not made it. This is the image taken from somewhere whose reference is given here. Again, this is another image in which you can see the various parts of a compound microscope. Parts of compound microscope may be divided into mechanical parts and optical parts. Mechanical parts include base or stand, pillars, inclination joint, curved arm, body tube, draw tube, resolving nose piece, coarse adjustment, fine adjustment, and stage. While optical parts may be classified as light source, iris diaphragm, sub-stage condenser, objective uh, lenses are fixed here, which are more than one in number. So first is scanning objective having power of 4x, Then low power objective 10x, high dry objective is of 60 and 40, and finally oil immersion objective of 100x power. And finally, last part of the optical part is eyepiece or ocular lenses. Now, light source is used as the source of illumination. Transmitted light is used in all types of light microscopes. It passes through the object and image is captured by the lens system. Light source may be sunlight or artificial light. Mirror and lens. The mirror reflects the light when sunlight is used as the light source. Light source other than sunlight requires both mirrors and lenses. As the mirror increases the light intensity, the lens brightens the light. Lens collects the light rays coming directly from the light source and also reflects the rays for convergence. Two types of mirrors are used. Plano mirror, which reflects the parallel beam of light for focusing of, there is no focusing of light. Then second variety is concave mirror. The reflected light is converged by the mirror. As a result, a cone of light is formed at the apex of the cone. The intensity of the light is maximum. When a condenser is used, plano mirror should be used. Use of concave mirror along with condenser system makes the condenser system inefficient. That is, when condenser system is not used, concave mirror is preferable. Condenser system. It directly facilitates the resolving power of the microscope. Resolution is achieved by optical means. During resolutions, aberrations that occur are minimized or ruled out by the process called optical correction. The condenser system is located between mirror and stage, and hence it is also called sub-stage condenser. It can also be located between the focal length and the stage. Its function is to collect, converge, and regulate light rays coming from light source on the specimen. Iris diaphragm. Beneath the condenser is an iris diaphragm regulating the quantity of light entering into condenser. It helps to obtain bright light from light source. Specimen stage. It is the square or round type. The square type of stage may be immovable or movable. Immovable stage possesses clips to hold the slides. Movable stage can be moved with the help of gliders. It can be moved forward, backward, and sideways. Round stage is run in polarizing microscopes. Now, objective lens system is located at the lower end of the tube facing the object. It is attached to resolving nose piece. It is very important as it affects the quality of image seen by the observer. The functions of the objective lens system are resolution and magnification. Their magnification may be 10x, 40x, 60x, or 100x, out of which 100x objective lens is called oil immersion objective lens. Most commonly used medium for immersion is cedar wood oil. with refractive index of 1.5, which prevents dispersion of light rays. The lens must be dipped properly and completely in the oil for accurate image formation. Focus adjustments. Accurate focusing is obtained by a special screw appliance known as a fine adjustment. Coarse adjustment knob is also present. Eyepiece or ocular lens. This is a short tube with two lenses which fits into the upper part of the microscopic tube. It is called the second lens system. Its main function is to magnify image formed by the objective lens. Eyepiece are marked 5x, 10x, 15x, etc. Most commonly, 10x Hygenian eyepiece is used in compound microscopes. It only magnifies but does not resolve the image. So, this was the explanation or description of variety of parts of microscope you are seeing here. This is the ray diagram of compound microscope, which means how the two lens systems make image and magnify it too. You can see that object is kept here behind the focal length of the objective. Its image is forming here within the focal length of the ocular lens, and the image formed is here, which is inverted and very much big or magnified. This is the real image and this is virtual image. Magnification of compound microscope may be calculated by this formula. We have already studied this formula. This is M. M is equal to D by FO multiplied by capital L divided by FE. D is the least distance of distinct vision, that is 25 centimeters. L is the length of microscopic tube. FO, focal length of objective lens. FE, focal length of eyepiece lens. Objective lenses, more about objective lenses. You can see here, these are mounted on nose piece. It forms magnified real image and magnification of objective, as you have already seen, may be divided by optical tube length, may be divided by focal length. And types of objective lenses are scanning lens, low power lens, high power lens, and oil immersions. These objectives are fixed on rotated nose piece. This immersion objective lens is marked separately by a black ring line indicating that this is immersion objective lens. Each lens has its details written on this part of the lens. Oil immersion objective, how it works, you can see that these lights are coming from the specimen. Then this is glass. Again, when light comes out after striking the specimen here, though this light is going out, this and most of the light rays enter objective lens because it is in close proximity with the specimen, and here we put oil. Oil prevents refraction of light outwards and allows it to pass straight into the objective, and it gives highest magnification of the specimen. You can see A, B, C and D are in air, while F, P, E and G are in oil. So this is used for making image. Now eyepiece eyepiece forms magnified virtual and erect image. It can be monoocular or biocular or tri trinocular. Another classification may be Hygenian eyepiece, Ramsden eyepiece, and compensating eyepiece. Total magnification which we get from the microscope, power of objective lens is multiplied by the power of eyepiece lens, and total magnification we get in each case. We can see here, this is if it is 4x, then it will be 40x, 10x, sorry, while 10x make 100x, 40 multiplied by 10 is 400, and here 60x gives 600. If it is 100x multiplied by 10x, then it will be 1000x, which is the total magnification we get by oil immersion objective lens. And I think this is all for this present lecture. Let me check if I have left anything. Okay, I have covered all the slides present here for today's lecture. One more important thing, that I have used some figures from other resources, and so this is purely for the lecture of for my students, and this lecture is not for private use, because I'm using some other resources also, and taking their pictures. I have not made those pictures myself, so it is only for my students, and I'm closing down my lecture now. Thank you.